Now let us continue with the two species of sugarcane. One is Saccharum officinarum, another one is Saccharum barberi. So here the Saccharum is nothing but what we are talking about, the varieties of sugarcane. We are discussing about the two important varieties of sugarcane. So here genus is same, that is Saccharum, but species is different. One is officinarum and other one is barberi. Now if you look at Saccharum officinaris is the one most commonly grown in case of South India, whereas Saccharum barberi is the one which is grown in North India. So we are taking the two varieties. The two varieties are grown in two different places of across India. Most commonly the South Indian variety is Saccharum officinarum and North Indian variety is barberi. Now if you look at the characteristics, for Saccharum officinaris, if you look at, we find a thicker stem and high yield, high sugar content. Means here you find very thick stem. When you find thick stem, definitely you find high yield and you find more sugar content, but one drawback here is this particular variety cultivated, grown only in South India. Why? The plant does not have ability to grow in North India. It cannot be cultivated in northern part of India. It can be cultivated only in south part of India. Why? The plant does not have ability to grow in North India. Right. Then when this particular plant taken as one of the parent, it's been crossed with another parent. The parent is Saccharum barberi. So here you find here it contains poor yield, means it produces very less sugar content, very low sugar content, very low yield will come. And remember, if you imagine this plant is growing in north part of India. Now when these two plants are crossed here, we get the plant, we get the sugarcane plant or the offspring. Generally, we expect the offspring have the combination of characteristics of both the parents. Now, this particular offspring has ability where it has produced with a high yield. High yield means here, obviously we talk in terms of sugar. That means we find high sugar content. And if you look at the stem part, the offspring has the dominant character, thicker stem, and most importantly it achieved, so I am keeping the star marks for the characters. Here it got the ability, it could able to grow, it could able to grow, where do you write completely? Now it got the ability, it could able to grow in North India. So what exactly meaning of this? Guys, here two characters have been combined. One is having high sugar content, other one is having low sugar content. When these two are combined, we got the offspring or the daughter plant having a good characters. What are the good characters? A dominant character it has a thicker stem. When thicker stem is there, guys, always it's not ensured that thicker stem will give high yield. No. In some of the sugarcane varieties, even though thick stem is there, but we find very low sugar content. But the offspring here has got with all desirable characters, thicker stem, and it is producing higher yield. Obviously it has high sugar content and could able to grow in north part of India. The plant got ability to grow even in colder regions. South India temperature little bit high, and compared with all seasons, average temperature will be little high when compared with the north part. But the genetically modification is done when two plants have been crossed. Resultant one has got the ability to grow in north part of India. Now this offspring has got desired with all required characters. Now similarly, let us talk about, even we can talk about millets, or you can take pulses. Millets nothing but you can take pulses and cereals. Now, when we make the millet plants to have the process called as a plant breeding. Now, with this plant breeding mechanism, we could able to produce the plants with the desirable characters. What are the desirable characters? So, the plants definitely they are no first requirement is met, that is getting a high yield, and the plants could able to grow. Plants are able to grow even in water stress environment, even in water stress conditions. Okay, so when we take the millets here and the process called as plant breeding, no doubt we are getting the varieties which can produce more tons quantity of the yield, and along with them even you can find the plants growing in water stress environment. What does it mean? Now my millets, my pulses can be grown absolutely if there is no rainfall, but still no problem, they can grow and cultivate. Suppose the drought condition is existing, still they can grow, very adequate amount of groundwater is there in the soil, or no groundwater also, but still my plants which are produced by the process called plant breeding, definitely they are giving a high yield and they could able to resist water stress environmental conditions. So, these are the good varieties with the desirable characters are made it possible with the process called as what? Plant breeding. Now, let us continue with the major process. So, from that time we are discussing many examples. Now, let us continue with plant breeding. So, if you look at the plant breeding. So, guys, if you look at here so far what all examples we have discussed here are mainly because of the process called plant breeding. Plant breeding concept is very important. Now, let us discuss about how the process of breeding can be done by using two plants or maybe the combination of genes. Now, let us discuss very important concept called plant breeding. Now, let us talk about plant breeding. In this, various steps are there. Steps or the mechanism we are going to study. The first one is called as collection of variability. So, we have very first step, it's called collection of variability. So, what exactly meant by variability? Variability means variations. So, initially we collect wildlife. So, we can take wild plants. Guys, we are discussing plant breeding concept. How to cross two plants to produce a better one with good desirable characters. How it is can be done, we are discussing now the process. In the process or the mechanism includes various steps. The very first step is collection of variability. So, in nature we find natural varieties. For example, I am going to go for plant breeding methodology for producing a better quality of rice grains, or maybe a better quality of fruits, or maybe better quality of vegetables, right. So, for that what do I need to do? First one, I have to see different variations in the plant. For example, if I take a rice plant, one particular rice plant growing in one area may have a bigger size. In some areas thin grains we get, in some areas highly nutritional, some areas good quality but not highly nutritional. So, different variations are there, different variety of plants are grown across India in different parts, in different regions, in different places. First of all, I have to see all variations. For that what do I need to do? I need to collect different plants with different genetic variations. For that we need to take what are called wild plants. What are wild plants? Naturally growing plants. So, I take for example the rice plant, what I find from Andhra Pradesh, definitely whose characters and whose quality, composition of the grain, size, color, texture of the rice grain, what's been cultivated in one part of India like Andhra Pradesh or Telangana, definitely it will be different from the rice grain what is being cultivated in north part or in Maharashtra or Bihar and all that. Okay, so the quality or the appearance or the quantity and the yield of one particular plant, definitely it is affected by surrounding environment. So, I will collect all the wild varieties, all wild varieties. So, when I collect all wild plants, what do I need to do? So, for example, this is plant A and this is plant B, right? Like there so many plants I have collected like this. Maybe when 10 plants or 100 or thousands of plants have been collected, maybe my first plant has high yield is there, or maybe it shows what you call very tall rice grains. Let me have very tall rice grains. I do not want this plant. Right. Second one, maybe the rice grain is very tasty, highly nutritional. Maybe I want the second grain. How do I come to know that? I'll take the cell or the seed of this and I'll cultivate the plant and I'll grow the next generation in front of me, and when I'll get the next generation, and I have to be very patient until I get the yield and to see the quality of the grain, I have to wait. So, then after I see that every plant has a different character. Guys, no need to worry anything. What I am telling you, it's very, very simple. Try to concentrate, try to understand this. Nothing. I want to produce a better quality of rice, maybe a better quality of vegetable. Or as I told you for the last class video, I want better quality of coconut. For that, available type of all rice plants or available type of all coconut plants I will collect. For example, A plant is a rice variety from Maharashtra which I collected. Maybe B plant is rice variety from Tamil Nadu or Chennai. C plant is from Bombay, or D plant is from AP. Like that, different varieties are taken. Right. And from there what we do? We take the seeds. When all seeds of different varieties are placed in the culture, this is what is called germ plasm collection. This is what is called germ plasm collection, right. So, when you take this here, germ plasm collection means what? We are taking the seeds of different varieties. I hope everyone got it. This is very simple topic. Concentrate and listen. I repeat again. Collection, we are collecting. Variability means variety of seeds. Variety of seeds. The seed is collected, from them plant is produced. So, it is called germ plasm collection. So, seed contains the cell, seed contains all the cells, seeds contain protoplasm. So, from the basic wild varieties we are collecting the seeds, it is nothing but collection of germ plasm. Now, what do I do now? After taking the seed really giving the same characteristics what I observed from the parent or not, again I will put it in the soil, I will grow the plants and I will see the characters. First plant is giving bigger size of the rice, second plant is giving highly nutritional, third plant is having high protein content, fourth plant is having disease resistance. Like that, every plant has its own character, its own good character. So, collection of all varieties. When I want to produce anything new, first of all I need to have collection of all different varieties of that particular plant. That is the first one called collection of variability. Right. Next, second one is we can take selection of parents. One second. So, what is the second one we have? We have is nothing but selection of parents. First I collected all plants, different varieties. Now I need to select the parents. So, again I will see that all plants are in front of me. There are different variety of plants are there. A plant, B plant, C plant and D plant. There are different variety of plants are there. Maybe the first plant has the character called high protein content, and maybe the plant D has disease resistance. Okay. Again I repeat, first plant is having high protein content, means quality wise, nutritional wise, maybe it is a good quality of the protein what we find, it has high protein content. And the last one may be disease resistance, it show resistance to diseases. I want these two characters. What I want? I want these two characters, high protein content and disease resistance. Second plant may be having bigger size of the grains. Third rice plant is growing very tall and giving short rice grains, but giving more yield, but still I do not want those characters. Out of all parents of wild plants which I collected, I selected two plants to cross. One of the plant is having high protein content, other plant is having disease resistance. Now I select these two plants as parents. It is called as selection of parents. Next character is cross among or cross or hybridization of selected parents. Okay, cross or hybridization of selected parents. Now if you take the cross or hybridization of selected parents, what do we do now? Let us take plant A, let us take plant B or D we can take. Now these two plants, one is high protein, another one is disease resistance. Now what do I have to do now here? It should be multiplied. It should be multiplied. So this is parents, they have taken it as a parents. When you make these two parents, we need to multiply. But before doing the multiplication, let me tell you one thing very clearly. Let us go for hybridization. What happens in hybridization here? In the process called hybridization, one plant having a flower taken it as male parent, right, one plant is taken it as what? Male parent, and other one is taken it as a female parent. Okay, one is taken as male parent, other one is taken as female parent, right. So here from this pollen grains are collected, a desired pollen grains are collected, and we transfer pollen grains from anther to stigma. We collect the pollen grains from anther, we get it transferred to stigma. So on the surface of the stigma, when pollen grain is dusted, the pollen grain enters inside and fertilizes with the egg. As a result, there is a formation of zygote. Zygote develops into embryo. Embryo develops into what? Seed. Zygote develops into embryo. Embryo develops into seed. The seed gives rise to a plant having both characters. So this is a F1 plant, or we find this generation is called F1 generation. Now this particular daughter plant or the offspring plant, this particular offspring has combination of both characters. What? It shows high protein content as well as disease resistance. High protein content as well as disease resistance. That means here offspring plant what is produced, there may be some hundreds of offsprings are produced, maybe in 100, maybe 10 in 100 or 1 in 10 or maybe what you say 100 in 1000 plants are produced with desirable characters. That means in F1 generation, that means if you get produced with 10 plants, in that one plant shows desirable combination. Suppose in F1 generation you produced with 10 plants, but in that hardly one plant have chance to show desirable combination. Why not remaining 9 will not show desirable characters? They show the combination of some other characters. Here first one is with a desirable character. Remaining 9 may be high protein content, but they may go for giving a bigger size of the grains. Maybe another plant is having disease resistance, but it shows very short grains and no proper nutrition. Like that, different genes with different genetic combinations are possible. So when you cross 2 plants, there is no guarantee that all 10 plants will give what I want the desirable character. No. 10 plants shows 10 different combinations. That is why we generally say in genetics, no, all 4 sons of the same father and mother may not be the same. Every son has his own characteristics, right, may not be the same. Why? Because different genetic combinations. So all 4 sons will have 4 different genetic combinations. Similarly, in F1 generation means first plant, first hybrid plant produced by crossing 2 plants. This plant, 10 plants are produced, all 10 plants are not same. In the 10 plants, maybe my desirable character is there in any one of the plant. So that is the one plant what I want. I don't want remaining 9 plants. I want only one plant or few plants which show my desirable genetic combination. How do I come to know that the plant is produced with a desirable combination? I will take the seed, I will sow it into the soil. I have to be patient. As a scientist, we have to be patient until the plant grow and grains are produced. Then we have to look for whether desirable combination is produced or not in that particular daughter plant, in that particular offspring. That is called F1 plant, or that is called F1 generation. Right. Next I will continue with, again I will take from F1 seed. From this plant I will take the seed, again I will put it in the soil, I will grow F2 plants or F2 generation. F2 plants or I will grow F2 generation. Okay. So if I take F2 plants and F2 generation, why I am taking again? From the all F1 plants, I took the seeds and I put it into the soil. I waited until F2 plants to grow. In F2 plants, again the same thing. All 100 plants may not have, hardly 10 plants will, out of 100 plants, maybe 10 plants have my desirable combination. So here there are so many plants I am growing. These all are F2 plants. These all are F2 plants. Maybe this show desirable character, maybe this show desirable character, maybe this show desirable character. Like that, if I count, hardly 10 plants may have desirable characters. Right. Again I will take the seed from this. Again I will put it in the soil. Like that, F3 plants will come. From them F4 plants will come. Like that, for number of generations we go for what is called multiplication. We go for what is called as multiplication, and this is multiplication itself is called as the third important step, that is nothing but selection of superior, or fourth important step, that is testing and selection of superior recombinant. but I will write here. So from here it is a fourth step, selection and testing of superior recombinant. Okay. What is that? The total story what we have discussed here. The total story what we have discussed here will come under the fourth step. Why it will come under the selection? From these crossing these two plants we got so many seeds. We sow the seeds into the soil. We got around 100 plants and that 10 are of our desirable characters. We are selecting. Right. And again testing. From this we are taking the seed. Again testing and putting into the soil. We wait until second generation to go. In that how many plants are having desirable characters, we are testing again. From them again we are taking the seed, putting into the soil, growing for next generation. We are testing how many plants are having the desirable characters. Now this how we can do here. Now we have to do here, in every step we have to do self-pollination. In every step we have to do self-pollination. What is self-pollination? If you take F2 plant, because these F2 plants are bisexual. Bisexual meaning what? Both anther and stigma are located on the same. Anther is here, stigma also here. Both anther and stigma are located on the same flower. So there is a transfer of pollen grain from anther to stigma, so that it will get fertilized, egg will come and embryo lead to formation of seed. Right. What is this? This is F2 plant. From this what you get is nothing but F3 plant. In F3 plant also we go for again self-pollination. Why self-pollination is done? Here until to reach homozygosity. Until to reach homozygosity. I hope understood everyone until here. So what I have written here? Right. Now listen carefully here. F1 plant is giving rise to F2 plant. F2 plant is giving rise to F3. F3 is giving F4. Like that, so many generations we are growing the plant. Every step we are doing self-pollination. Why self-pollination means? Until to reach homozygosity. What's meaning of homozygosity? Now you took some F5 plant. F5 plant with a desirable combination from which a cell is collected, a plant cell is collected. Same story. It has nucleus, plant cell has nucleus. Within that we find DNA is here. DNA contains gene. Now on the gene if you look at, this is DNA. On the DNA you find the gene. Each gene contains 2 alleles. You will come to know this later. Each gene or one gene contains 2 alleles. Guys, try to understand carefully. In genetics we will come to know later what exactly meant by alleles and all that. At that time you will get better clarity, but now presently it wasn't there here, but you have to understand the process. I am telling the part of the genetics. So don't worry, this we will discuss in genetics. Try to understand carefully. So here on DNA, each gene contains 2 alleles. Now one allele is called dominant allele. What's the meaning of dominant allele? It expresses for generations together. Other one is called as recessive allele. Other one is called as what? Recessive allele. So what's the dominant? For example, if you take height of the plant. If you take height of the plant. So when you take the height of the plant, the dominant allele gives you the tallness here, and recessive allele gives the dwarfness. What's the meaning of that? If the plant is tall, then it is called dominant allele got expressed. If the plant is tall, it means that dominant allele got expressed. That means at that time recessive allele kept quiet. Means dwarfness causing gene became inactive, allele became inactive, and dominant responsible allele got active. That is why the plant is tall. In some cases, the plant is dwarf. Means recessive allele got expressed and dominant allele got inactive. That means every gene has control of 2 alleles. The character which expressed generations together is dominant character. The character which may express few times in the generations may not express, it's called recessive allele, right. So here in this case, when I say here, if both are dominant, then we say allele capital T capital T. Capital T capital T means this is homozygous. Homozygous means if this allele express also tallness will come, this allele express also tallness will come, right. That means both alleles are same, it's called homozygous. So you are going on self crossing until to reach homozygosity. Sometimes you find alleles like this, capital T, small t. That means the condition is heterozygous. The condition is what? Heterozygous. If this allele expresses, it will become tall. If this allele expresses, it become dwarf. This expresses means it will become tall. If small t allele express, it will become dwarf. That means this is called heterozygous condition. Suppose our F3 plant is heterozygous condition. Maybe one of the plant may be tall and other offspring may be dwarf. So we don't want chances, we don't want to take the chance. We want all plants coming out of this particular F5 plant, all should be tall. That means it should be in homozygous condition. Guys, if in case if you don't understand what is called gene or alleles, dominant allele, recessive allele, not a matter, because we are going to discuss in genetics. Actually this first part was not there in your plant breeding. To make you all understand so clearly, suppose in your little mind the question arises, why we need to take so many generations? Because it is not a simple process. Farmer will collect the seed, F1 seed, he will put it into the soil, scientist will put it into the soil, F2 plants will grow, it takes lot of time, and after F2 plants grow, in that how many plants are of parent type we have to identify. And 10 in 100, out of 100 F2 plants, 10 plants resembles F1, right. Now we take 10 plants and from this seed is collected, again we go for F3, right. Why we need to do this much? Because when you keep on multiplying the plant, it is very a time consuming process, time consuming and very tedious process. Okay, takes generations together, it takes a lot of time, and moreover it is tedious, complicated process, right? Why do we need to wait for so many generations? What is the need? You may get doubt, right? You may get a think that why do we need to collect so many generations together, taking the seed, putting in the soil, getting F3, again from we have to wait until F3 plants grow, see how many of them are visible, F2, those plants you need to select, again collect the seed from them, put it into the soil, F4 will come. Out of 1000 plants of F4, all may not have desirable characters, maybe 100 plants. From them collect the seeds, go for F5. Why do we need to do all these things? Because until to get homozygosity, you need to go for what? Self-pollination. You need to go for self-pollination for several generations together until to get homozygosity. What happens when homozygous condition comes here? From that no need to worry. Whatever the plant comes from this, it will be tall only. If the plant comes from this allele also, tall only. That means all plants will be produced with a desirable character. So this allele express also the same plant will come. This allele express also the same plant will come. This allele express also we fed high protein content. This allele express also high protein content. If it is for one gene having one character called, if this gene responsible for high protein content, no matter this allele also high protein content, this allele also high protein content. That is the need for us to go for self pollination until to reach homozygosity. Right. Next is we take commercialization, or we say testing